Welcome to the Deep Dive Learner. So, I want you to imagine holding this tiny glass vial in your hand.
You're right. And we're talking about a very, very expensive vial here.
Oh, exactly. It's worth roughly $1 million. And inside is this custom-engineered biological cure, right?
Yeah.
It's perfectly designed to target disease cells in your body without harming anything else.
Which is, the absolute cutting edge of medicine. It's what we've been working toward for decades.
It is. But now I want you to imagine that simply sliding that vial across a cold metal counter or like gently tapping against your palm completely destroys the medicine inside.
I mean, it sounds like a massive exaggeration, right? But that is the invisible reality that clinical researchers are dealing with every single day right now.
Yeah.
We're talking about therapeutics that are these evolutionary marvels, but they're essentially trapped inside a microscopic house of cards.
Right now, the entire medical world is talking about exosomes. They're being hailed as the revolutionary future of regenerative medicine and targeted drug delivery. If you're not familiar with them, think of exosomes as nature's perfect microscopic cargo ships. They're these incredibly tiny lipid-bound bubbles that our cells use to send potent biological messages.
Yeah, things like RNA and specialized proteins, just passing them to one another.
Exactly. And inside the human body, they are incredibly robust. I mean, they navigate the turbulent, rushing rivers of our bloodstream, they cross dense cellular barriers, and they deliver their payloads with absolute perfection.
Well, yeah, inside the body, they have the benefit of billions of years of evolutionary engineering, right? The environment is perfectly calibrated for their survival.
Right. The temperature, the pressure.
Exactly. Blood viscosity, temperature, pressure, it's all highly regulated by the body.
But the moment you take those perfect little cargo ships out of the body to, turn them into a clinical treatment, everything changes. The hype is all about the biology, but nobody outside the lab talks about the physics.
Which is the real bottleneck.
Right. Just moving these exosomes around a clinic, or letting them sit in a standard plastic tube, or pushing them through a syringe can just obliterate them before they ever reach you, the patient.
It's wild. We spend millions of dollars and years of research isolating and purifying these therapeutics. Yet the artificial environments of our clinics, the plastics, the temperature shifts, the physical forces of everyday medical tools, they turn out to be a lethal obstacle course.
A lethal obstacle course for these delicate lipid bilayer vesicles.
Yeah. The biology might be perfect, but if the physical delivery mechanism fails, the treatment is useless.
Okay, let's unpack this, because our mission for this deep dive is to look at the meticulous, almost paranoid precautions that clinical staff have to take just to keep these exosomes alive.
Paranoid is definitely the right word for it.
Oh, for sure.
Yeah.
We are pulling from a stack of specific scientific studies, handling protocols, even fluid dynamics research to understand exactly how we get these fragile medicines from a laboratory freezer into your body intact. We're looking at the how and the why behind the survival of the world's most delicate medicine.
And that journey really starts before any active medical procedure even begins. Because before an exosome therapeutic can be prepped for you, the patient, it usually just has to sit in a container.
Right.
And that introduces us to the very first highly unexpected enemy in this clinical obstacle course, which is the storage tube itself.
Yeah, I was looking at this staggering statistic from a PLOS1 study in our research stack, and it honestly blew my mind.
The 48-hour one.
Yes. The researchers took a batch of highly purified exosomes and put them in an ordinary 2 milliliter polypropylene tube.
Which is just your standard everyday lab tube. They're everywhere.
Right, cheap plastic. They left them in the fridge at a standard 4 degrees Celsius. And after just 48 hours, they measured the concentration, and they had lost 51% of the exosomes. Over half the medicine just gone.
And the crazy part is the medicine didn't degrade chemically, and the tube certainly didn't have a hole in it.
No. I picture it like trying to pour raw, thick honey out of a glass jar. You can hold it upside down all day, but half your honey is just hopelessly stuck to the inside of the glass.
That analogy actually captures the end result perfectly. At a microscopic level, we are dealing with a phenomenon called diffusion-controlled adsorption.
Diffusion-controlled adsorption. Okay, break that down for us.
So to understand why this happens, we have to look at the chemistry of both the exosome and the plastic. Exosomes are enclosed by a lipid bilayer, right?
A fat layer, essentially.
Exactly. And lipids have regions that are hydrophobic, meaning they actively repel water. Now, polypropylene plastic, the material most standard lab tubes are made from, is fundamentally a nonpolar hydrophobic polymer.
So it's essentially a magnetic attraction between two things that absolutely hate water.
That's a great way to put it. In a water-based clinical buffer solution, those hydrophobic areas on the exosome membrane are thermodynamically driven to seek out other hydrophobic surfaces.
Just to get away from the water.
Right, to minimize their contact with it. So as these tiny lipid vesicles bounce around in the liquid due to natural thermal motion, what we call Brownian motion, they inevitably bump into the walls of the plastic tube.
And when they hit the wall,
When they do, those hydrophobic interactions cause them to bind tightly to the surface. They stick, and they do not let go.
and the physics of how they pack themselves onto that plastic wall is wild. It's not just a random, messy scattering, is it?
No, not at all. The researchers detailed the theoretical math behind the binding. As the particles adhere to the curved wall of the tube, they naturally arrange themselves into this highly efficient pattern to maximize space.
Right.
They form a hexagonal packing arrangement.
It's like trying to cram as many oranges into a wooden crate as physically possible. They naturally settle into the little gaps between one another.
Yeah, exactly. They pack themselves tightly and uniformly along the entire inner surface of the tube until the binding capacity of that plastic wall is completely maxed out.
Oh.
And if the total number of exosomes in your therapeutic solution is relatively low, which is, very common with these highly purified targeted medicines, that geometric packing on the tube wall can literally swallow up the majority of your dose.
Because the plastic surface area is just vast compared to the nanoscale size of the exosomes.
Vast. It's like a massive parking lot for microscopic cars.
Okay, I have to ask the obvious question here. If we know that cheap plastic is literally eating half of our $1 million medicine, why on earth are we using it? Are we really trying to mass produce the equivalent of microscopic Fabergé eggs and just tossing them into cheap Tupperware?
Well, the industry is having that exact realization right now, thankfully, and protocols are rapidly changing to address it.
Okay, good.
The sources reveal two main biochemical solutions that clinics are adopting to outsmart the plastic. The primary solution is swapping out those standard polypropylene tubes for what are called protein lobin tubes.
Protein lobine, got it.
These are specifically engineered plastics that have been modified to have a highly hydrophilic or water-loving surface.
So they flip the magnetic polarity, essentially. By making the wall water-loving, the water-hating parts of the exosome have no interest in sticking to it.
Exactly. The physics completely change. When the researchers tested these lobine tubes under the exact same conditions, the exosome loss dropped from 51% down to just 18 to 21%.
That is a massive preservation of the dose simply by altering the surface chemistry of the container.
It really is.
But, those engineered tubes are expensive, and I imagine supply chain issues exist. What happens if a clinic simply doesn't have them on hand?
There is a brilliant chemical workaround called pre-blocking.
Pre-blocking.
Yeah, if you are forced to use a standard sticky tube, you can coat the inside of it before you ever add your medicine. The common method is using a solution of bovine serum albumin, or BSA.
Wait, albumin's a protein, right? How does adding cow protein to a tube save the exosomes?
So BSA is a relatively bulky, highly abundant protein that is notorious in biology for its ability to stick to surfaces. You essentially flood the empty tube with a BSA solution.
Oh, I see where this is going.
Yeah. The BSA rapidly undergoes conformational changes and binds to all those hydrophobic sites on the plastic. It acts like a decoy wave.
It paves over the sticky real estate.
Exactly. After a short incubation, you wash out the excess BSA and pour in your actual exosome therapeutic. Because the tube walls are already completely saturated with BSA molecules, there are no parking spaces left for the exosomes.
So they remain freely suspended in the liquid. That's so clever. But what's crazy to me about all of this is that the tube doesn't just lower the overall amount of the dose. It actually changes the fundamental nature of the medicine itself.
What's fascinating here is that this diffusion-controlled adsorption process predominantly causes the loss of the smaller particles in the solution.
Because smaller things move faster.
Right. Precisely. In fluid dynamics, smaller particles diffuse more rapidly. They are bouncing around with much higher kinetic energy compared to the larger, heavier vesicles.
So they hit the walls first.
Because they cover more distance in less time, they're statistically much more likely to hit the wall of the tube and stick first. This means if you leave your sample in a standard tube for a day, you aren't just uniformly losing half your dose.
You're losing a specific type.
You are entirely skewing the size distribution of the clinical sample. The liquid you pull out of that tube is physically and biologically different from the medicine you put into it.
Oh, wow.
The smaller exosomes, which might carry very specific crucial RNA payloads, are disproportionately gone.
Which is a complete nightmare for standardizing a medical treatment. I mean, you have no idea exactly what you're injecting into the patient anymore.
Exactly. It's a massive variable.
Okay, so let's say the clinic gets it right. They use the low-bind tube, or they pre-block with BSA. The medicine survived the fridge. But now, a human being has to physically pick up that vial and prep the dose.
Right, the handling phase.
We are moving from passive storage to active handling. And here's where it gets really interesting.
This is where human error intersects with fluid dynamics.
I was reading through this exosome reconstitution step-by-step guide from our stack, and the level of extreme caution it demands is almost comical if it weren't so serious.
It reads like a bomb defusal manual.
It really does. You cannot just grab the tube and shake it up to mix it. It's less like mixing up a scoop of pre-workout powder, and more like trying to merge onto a busy highway driving a car made of wet paper.
That's a terrifying image.
Right. The protocol literally mandates that the clinician must gently pipette the liquid up and down exactly 10 to 15 times. Not 5 times, not 50 times.
That exact number is derived from careful studies of membrane limits. The fragility comes right back to the physical vulnerabilities of the exosome's lipid bilayer.
What forces are actually at play there? Because the guide also strongly warns against using a vortex mixer. You know, that little vibrating rubber cup you see in labs that creates a fast whirlpool in the test tube.
Oh, yeah, the vortexer.
They say vortexing should strictly be kept to an absolute maximum of 60 seconds. Why is a tiny whirlpool a death sentence for them?
It comes down to severe shear stress. When you vortex a liquid or rapidly force it in and out of a narrow pipette tip, you aren't just moving the liquid as one solid block.
Okay, so what's happening?
You are creating layers of fluid that are moving at drastically different velocities. The fluid near the center of the whirlpool is moving much faster than the fluid dragging against the sides of the tube.
So if a microscopic exosome gets caught between a fast-moving layer of water and a slow-moving layer,
it experiences intense frictional forces pulling in opposite directions. The lipid bilayer is incredibly thin, only a few nanometers thick. If the shear stress exceeds the mechanical tensile strength of that membrane, it literally rips the lipid bilayer open. The exosome ruptures, spilling its valuable genetic cargo out into the surrounding buffer where it is instantly degraded and rendered completely useless.
it just shreds them. The protocol also repeatedly screams, "Avoid bubbles," in bold letters.
Oh.
I have to admit, I always thought of bubbles as harmless, maybe even a sign that something is well mixed.
To a microscopic lipid vesicle, a bubble is a violent environmental hazard.
Violent.
Absolutely. When a clinician pipettes too aggressively and creates foaming, they are introducing massive amounts of air into the liquid. This creates vast networks of air-liquid interfaces.
And at a microscopic level, surface tension is incredibly powerful, right?
It is an immense force on the nanoscale. The hydrophobic tails of the exosomes are drawn to the air in those bubbles. When those tiny bubbles inevitably pop or collapse, the rapid shift in surface tension forces creates a microscopic shockwave.
The shockwave.
It physically damages the membranes. Furthermore, the trauma of these forces causes the surviving exosomes to exhibit a stress response they aggregate.
They clump together.
They clump tightly together into large, useless masses.
And if they clump together, you've ruined the homogeneity of the dose. You might draw up a syringe and inject a patient with one massive, concentrated clump of exosomes, and then the next syringe is just empty liquid.
This raises an important question for clinical workflows. How do we guarantee standardization and safety when a clinician simply pressing their thumb a little too aggressively on a pipette plunger can fundamentally degrade the therapeutic?
I mean, my first thought is, why don't we just automate the whole thing with robots? Take the heavy-handed human out of the equation entirely.
Well, automation is certainly being explored for large-scale manufacturing. But at the point of care in a hospital room or a local clinic, human handling is still required.
You can't have a robot in every single exam room.
Right. Therefore, the sources outline a very strict physical regimen that clinicians must memorize. First, they must use specialized low-retention pipette tips.
Again, fighting that hydrophobic stickiness we discussed with the storage tubes.
Exactly. Second, they must master a slow, controlled thumb release to absolutely avoid creating any air-liquid interfaces or foaming.
No bubbles.
No bubbles. Third, if they need to gather the liquid at the bottom of the tube after mixing, they can use a microcentrifuge. But they are restricted to brief spins at very low speeds, typically around 1,000 to 3,000 times gravity.
Anything faster induces destructive G-forces, I imagine.
Exactly. And finally, the biological clock is ticking. The reconstituted product must be used within 2 hours.
Okay, so we've gently coaxed them off the plastic, and we've mixed them with the delicate precision of a bomb squad.
A very stressed bomb squad.
Right. But we've completely skipped over a massive environmental shock that happens before any of this. Because these therapies don't usually ship to the clinic sitting at a comfortable room temperature.
Definitely not.
They arrive deep frozen, usually packed in dry ice at -80 degrees Celsius. Or they arrive lyophilized, which means freeze-dried into a powder. Bringing them back to a liquid state at room temperature is arguably the most terrifying gauntlet of all.
The thermal shock phase is where the most catastrophic structural damage occurs if the physics of freezing and melting are not perfectly managed.
Think about what happens when you freeze and thaw a strawberry. It goes into your freezer, bright red, plump and firm. But when you pull it out and let it thaw on your counter, it turns into this sad, leaky, mushy puddle.
It's ruined.
What stands out to you, the learner, about how a microscopic bubble made of fat and water might handle that exact same freezing and thawing process?
It is a fundamentally brutal physical process for biological membranes. The sources detail that every single freeze-thaw cycle induces intense shear and osmotic stress on the membranes.
Because the water expands when it freezes.
Right. As the water inside and outside the exosome cools, it expands. As it thaws, it contracts. This constant flexing causes protein leakage from the membrane and rapid RNA degradation.
So it's squeezing the medicine out of them.
Every single time you freeze and thaw an exosome sample, you are permanently reducing its bioactivity by roughly 15 to 20%.
Wow. 20% of the medicine simply dies every time the temperature fluctuates.
Because as water freezes, it doesn't just get cold and stop moving, it fundamentally changes state. It forms a rigid crystalline lattice. Those growing ice crystals are jagged and sharp.
Like knives.
They act like microscopic daggers, literally slicing through the fragile lipid bilayers of the exosomes from the inside out.
Wait, if ice is the ultimate enemy here, why freeze them at all? Why not just keep them purely refrigerated as a liquid?
Well.
Oh, wait, we already answered that. If we keep them in the fridge, the plastic tube eats 51% of them. We are completely trapped by the physics.
It is a very precarious balancing act. To survive long-term storage, they must be frozen, but the act of freezing them is inherently destructive.
So how do we fix it?
The solution requires a combination of strict clinical workflow changes and advanced chemical engineering. On the clinical side, you absolutely must divide your master stock into single-use aliquots before the initial freeze.
Meaning you separate the medicine into tiny individual patient doses right at the manufacturing plant.
Precisely. That way, when you need a single dose for a patient, you pull out one tiny vial and thaw only what you need. You entirely prevent exposing the rest of your therapeutic batch to a second or third freeze-thaw cycle.
That makes total logistical sense. But when you do pull that single dose out of the -80 freezer, how do you get it back to room temperature without those microscopic ice daggers shredding the exosomes on the way up?
You perform what is known as a controlled thaw.
How controlled are we talking?
The strict protocol dictates rapidly thawing the vial in a 37 degrees Celsius water bath for 2 minutes or less.
That fast.
You want to force the sample through that dangerous transition phase where ice is actively melting, shifting, and recrystallizing as quickly as physics will allow in order to minimize thermal gradient stress.
You also mentioned chemical engineering. How do we alter the chemistry of the water to protect them? You mentioned lyophilization earlier, freeze drying. How does removing the water help?
Lyophilization involves freezing the sample and then placing it under a vacuum so the frozen water sublimates it, turns directly from a solid ice crystal into a gas, skipping the liquid phase entirely.
Okay, so no melting phase.
Right. This removes the water, leaving a stable powder. But removing the water creates a new problem. Water molecules naturally form a protective hydration shell around the lipid head groups of the exosome.
Uh-oh.
If you strip that water away, the lipids crash into each other, the membrane undergoes a phase transition, and it falls apart.
So we need something to hold the structure together when the water is gone.
Exactly. We use molecules called cryoprotectants. Compounds like dimethyl sulfoxide or DMSO can help. But the really fascinating breakthrough highlighted in the recent research involves a specific molecule called trehalose.
Drahulose, that's a type of natural sugar,
Yeah.
right? I've seen it listed as an ingredient in certain foods. How does a sugar stop an ice dagger?
It functions through a mechanism known as the water replacement hypothesis.
I like the sound of that.
When you add a high concentration of trehalose to the exosome buffer solution before freezing or freeze-drying, it does something remarkable.
Right.
As the temperature drops and the water molecules begin to freeze or evaporate, the trehalose molecules step in and form hydrogen bonds directly with the lipid heads of the exosome membrane.
Oh, they physically take the place of the water.
Yes.
They act like microscopic scaffolding.
Even more impressively, as it cools, trehalose doesn't crystallize like water does. It undergoes a process called vitrification.
Vitrification.
It turns into a highly viscous amorphous solid. It forms a literal glass matrix. It locks the exosomes in place within this solid sugar glass, entirely preventing the formation of sharp ice crystals and stopping the biological molecules from degrading.
It encases them in microscopic sugar glass. That is absolute brilliance.
It's an elegant solution.
Okay, let's recap this journey. We've got our medicine out of the deep freezer using our protective sugar glass matrix and a rapid 37-degree water bath. We've mixed the dose with our ultra-gentle, highly specific 10 to 15 pipette strokes without making a single catastrophic bubble.
Right.
We've got the liquid safely resting in a specialized low-bind plastic tube so it doesn't stick to the walls. The vial has survived, but now comes the final, unavoidable hurdle.
The injection.
Getting those exosomes out of the tube and into you, the patient. And to do that, we have to force them through a tiny, hollow metal needle.
The delivery danger. This is the moment where fluid dynamics becomes the ultimate gatekeeper of the therapy's success.
The sources dive incredibly deep into the fluid dynamics of syringe injection. They actually draw heavily on existing data from stem cell injections because living stem cells have very similar delicate membrane vulnerabilities as these cell-derived exosomes.
Yeah, the physical constraints are very parallel.
They also look at advanced microfluidics, and it all comes back to that physical enemy we met during the mixing phase.
Yeah.
Shear stress.
Exactly. When fluid is forced under pressure through a narrow, enclosed tube like a syringe needle, the liquid doesn't all move forward at the exact same speed.
I always picture this like rushing down a high-speed enclosed water slide at a theme park.
Oh, that's a good way to look at it.
If you are positioned perfectly in the middle of the tube, riding on the rushing water, you are flying. It's incredibly smooth and fast. But if you drift out to the edges and your skin drags against the plastic sides of the slide, the friction is brutal.
It is.
You slow down instantly, you get scraped up, and it's a completely different, much more violent experience than the person riding perfectly in the center.
That analogy perfectly illustrates the Hagen-Poiseuille principles and the physics formula provided in the clinical text.
Lay the math on me.
The shear rate during an injection is determined by the volumetric flow rate, meaning how fast the clinician is actively pushing the plunger divided by the radius of the needle cubed multiplied by pi.
The radius cubed.
Right. The mathematics dictate that the absolute highest shear stresses occur right at the metal boundary wall of the needle. The exosomes flowing through the exact center of the needle experience almost zero stress, but the ones trapped near the walls are experiencing severe velocity gradients that can literally tear them open.
Okay, let me play armchair engineer here.
Yeah.
If I look at that formula, the radius of the needle is cubed in the denominator. That means even a tiny change in the width of the needle makes a massive mathematical difference.
It does.
So logically, to protect the exosomes, a clinician should just use the widest, shortest needle possible. Just hook up a massive, short metal pipe to the syringe so nothing touches the sides. Right?
Yeah. If we connect this to the bigger picture, the logical math rarely translates perfectly to patient reality.
There's always a clinical catch. I suppose you can't just stab a patient with a giant pipe.
Patient comfort is certainly one factor, but the sources add crucial fluid dynamic nuance regarding the length of the needle. You are correct that a larger gauge, a wider needle, drastically reduces the immediate shear stress at the walls. However, different medical injections require different tissue depths. An intravenous injection requires a different needle than a deep intramuscular injection.
Right. You simply cannot reach deep muscle tissue with a wide, stubby little needle.
You need length, to reach the target.
Yeah.
And there is a punishing inverse relationship between flow rates and needle length.
What happens with the longer ones?
Longer needles inherently create much greater fluid resistance. If a clinician is pushing the syringe plunger at a constant speed through a long needle, the pressure inside the syringe barrel builds up significantly over time.
Oh, I see.
That prolonged compounding pressure can be just as damaging to the exosome membranes as the immediate friction at the walls.
It's exactly like putting your thumb halfway over the end of a running garden hose. The water sprays out faster, but the physical pressure built up immensely inside the rubber hose behind your thumb.
Precisely. Therefore, optimizing these injection parameters requires a delicate, highly calculated balance. Clinicians cannot just default to a wide needle because the anatomical needs vary.
So what can they control?
They must meticulously control the flow rate, literally retraining the muscle memory of the speed at which their thumb depresses the syringe.
Retraining their thumbs.
Yes. Furthermore, cutting-edge clinical setups are now utilizing microfluidic principles of gentle separation.
Gentle separation.
These involve specialized syringe tapers and tubing designs that naturally funnel the exosomes into the low-stress center of the fluid channel before they enter the needle. It actively minimizes their exposure to the harsh forces at the metal walls.
So what does this all mean for the future? We started this deep dive looking at exosomes as the undisputed magical future of targeted medicine. But what we've discovered is a terrifying clinical gauntlet of physical vulnerabilities.
It's a miracle they survive at all.
Seriously. Protecting these microscopic cargo ships isn't just about understanding the biology of the payload. It is entirely about mastering the physics of the delivery.
Completely. It requires swapping out standard plastics for chemically modified low-bind tubes to defeat diffusion-controlled absorption. It means banning aggressive vortexing and outlawing microscopic air bubbles to prevent catastrophic shear stress.
In the sugar glass.
Right. It requires encasing the medicine in trehalose sugar glass just to survive the thermal shock of a freezer, and it means mathematically calculating the exact radius of the needle and retraining the literal speed of the doctor's thumb.
The overarching takeaway from synthesizing all of this research is that while exosomes absolutely hold the key to next-generation medicine, their success relies entirely on rethinking our most basic, everyday laboratory and clinical handling protocols.
Basics matter.
Stability is not an afterthought to be figured out later. It is the primary technical bottleneck of the entire field. If we do not respect the physics of the delivery, the brilliant biology of the drug is rendered completely irrelevant. Which leaves us with a pretty haunting final thought to mull over. If the simple, everyday act of pushing a liquid through a standard syringe, or just letting a vial sit in a fridge for a weekend, can physically tear apart the medicines of tomorrow,
Yeah.
it really makes you wonder, how many other brilliant microscopic therapies, maybe even world-changing cures, have completely failed in clinical trials over the last decade simply because we used the wrong plastic tube? Or a nurse pushed a plunger just 1 second too fast.
It is a sobering possibility that keeps structural biologists awake at night. I mean, the medicine might have been perfect, but the vessel failed it.
It is definitely something to keep in mind the next time you see a headline about a promising medical trial unexpectedly failing. I want to thank you, the learner, for joining us on this deep dive. We encourage you to keep questioning the little details, because sometimes the difference between a global medical breakthrough and a total failure is just a single air bubble in a cheap plastic tube. Keep diving.